Dear colleagues, good evening. I would like to begin by thanking the organizing committee for the Young Investigator Award. Before I proceed, I must acknowledge that our research has received funding by the European Union. The polycystic ovary syndrome, or PCOS, is the most common endocrine abnormality in young women. It manifests with acne and hirsutism, but also with oligoanovulation and subfertility. Not only is PCOS commonly accompanied by difficulty to achieve conception, but even when pregnancy is accomplished, there is increased risk for adverse outcomes, such as gestational diabetes, hypertensive disease, and preterm delivery. Over the last decade, there is growing scientific interest regarding the role of the placenta in the polycystic ovary syndrome. Available evidence demonstrates that the placental structure is altered in PCOS. Studies have also shown that the activity of certain steroidogenic enzymes is altered in PCOS placenta in a way that promotes increased androgen production. These findings are very important for two reasons. First, they might be the key to explain the increased incidence of adverse pregnancy outcomes in PCOS, as defective placentation is a common denominator in all major pregnancy complications. Second, they could help us in understanding the disease's pathophysiology. According to the developmental theory, intrauterine overexposure to androgens creates an adverse environment which affects via epigenetic mechanisms the expression of several fetal genes, leading to a reprogramming of the reproductive and metabolic axis. This theory is supported by extensive experimental data in animals, which have highlighted the importance of placental alterations in the development of this adverse hormonal and nutritional intrauterine environment. Neurokinin B, or NKB, is a decapeptide belonging in the tachykinin family of neuropeptides. In humans, it is encoded by the TAC3 gene. Neurokinin exerts its actions by activating G protein-coupled receptors, the neurokinin receptors 1, 2, and 3. Traditionally, NKB has been classified as a neurotransmitter, which is produced along with kisspeptin and dynorphin by the K and the Y neurons of the hypothalamus. Their main function is to regulate the secretion of GnRH. However, neurokinin B is also produced outside of the brain. It has been demonstrated that NKB is produced by the placenta, and this is why there is a significant increase in its concentrations during pregnancy. The role of neurokinin in normal pregnancy has not been fully clarified. However, it seems that it is involved in the regulation of blood supply to the uterus, as it can have vasodilative as well as vasoconstrictive effects. Increased neurokinin production has been associated with several pregnancy complications, such as preeclampsia, intrauterine growth restriction, and preterm labor. In PCOS, although there is increased frequency of defective placentation and of complications such as preeclampsia and preterm delivery, the role of neurokinin B has not been investigated. The aim of our study was to compare the placental mRNA expression of neurokinin B and its receptors in pregnant women with PCOS and controls. This was a single-center, prospective, case-controlled study in which we studied 10 women with PCOS and 10 healthy controls with spontaneous conception and singleton uncomplicated term pregnancies. The placental samples were collected within 15 minutes of the delivery of the placenta. We obtained full-depth samples from 3 different areas of the organ. The maternal decidua and chorionic plate tissues were removed, and each sample was further divided in pieces of about 1 cubic centimeter. The sample tissues were then submerged to RNA later solution and stored at -20 Celsius until the analysis. For gene expression analysis, total RNA was isolated from the samples and used to synthesize complementary DNA. cDNA was then used for real-time PCR with specific primers for the genes of interest and SYBR green as a dye. The relative mRNA expression of NKB, NK1, NK2, and NK3R was estimated by the delta-delta-CT method using beta-actin as the housekeeping gene. Now let's have a look at our results. As we can see in this table, there was no significant difference between women with PCOS and controls regarding their mean age, their BMI at delivery, their gestational age, the mode of delivery, and the sex and birth weight of the offspring. In this figure, we present our findings regarding the mRNA expression of neurokinin B. The expression level in controls is considered as reference, therefore the relative quantification in the control group has a value of 1. We found that the placental neurokinin B expression in women with PCOS was 2.4 times higher than in controls, and this was statistically significant. Here, we can see our results concerning the mRNA expression of neurokinin receptors 1, 2, and 3 in the two subgroups. NK3R expression was similar in PCOS and controls. The expression of NK2R seems to be higher in women with PCOS, about 4.5 times, but this difference did not reach statistical significance. Of note, this neurokinin receptor isoform showed very low levels of expression in both groups. Therefore, in terms of absolute levels of expression, the importance of this difference is questionable. Finally, the placental expression of NK3R was 7-fold higher in PCOS versus controls, and this was a statistically significant finding. In conclusion, our study showed that the placental expression of neurokinin B and the neurokinin receptor 3 is increased in women with PCOS. Of course, these findings are preliminary and based on a small sample size. Hence, expanding the number of participants is necessary in order to corroborate our findings. Should these findings be confirmed, many interesting research questions emerge. First, could neurokinin B represent one of the mediators in the placental alteration characterizing PCOS? And if yes, through which mechanisms? Is the placental expression of neurokinin and its receptors different in the different PCOS phenotypes? And does it depend on the hormonal or the metabolic milieu of the patients? And finally, since we know that increased NKB production by the placenta leads to increased levels of this peptide, not only in the maternal, but also in the fetal circulation, could NKB have a potential role in PCOS heritability? Thank you very much for your attention.